Well, we've made it to the end of the road. This is our last module. This week we're going to explore genetics. This is a huge field, of course, and we're going to be just skimming along the top of it. There's so much neat stuff that we can talk about, and I've made an effort, though, to try and pare it down and keep it fairly simple and straightforward. I know you're tired, and I know I'm tired too, so we're going to try and stick to the basics and not overdo it. The focus this week is to discuss basic Mendelian genetics and then explore some of the exceptions to Mendelian genetics. One of the most important concepts that I would like you to take away with you is the connection between genes, alleles, meiosis, gene expression, meaning the production of protein, and the role of protein in generating a phenotype. This is a big ask, I know, but you can do this, and it will tie together a number of different topics in the course. We're going to tackle those connections in the first lecture. This lecture is about Mendelian genetics, and Mendelian genetics is named after Gregor Mendel, the monk who conducted scientific experiments in the mid-19th century, trying to work out the rules or the principles of inheritance. His model organism was the pea plant. He chose pea plants because they were easy to work with and they came in different varieties that had simple and easy to identify differences in specific characteristics. For example, one variety always produced tall plants and another variety always produced dwarf plants or short plants. He referred to the height of the plant as a character, and whether or not the plant was tall or short as different traits of that character. And to give you some idea of the other kinds of characters that Mendel worked with, there were varieties that had produced round seeds, varieties that produced wrinkled seeds. Some varieties had yellow seeds versus green. There were differences in pod shape and pod color, and finally, differences in flower color as well. At the time that Mendel was working, there were two major hypotheses about the nature of inheritance. The first was the belief that offspring exhibited traits that were actually a blend of the parental traits. So, for example, if you were to cross a plant that produced red flowers with another plant that produced white flowers, you should see pink flowers in the next generation. Another hypothesis or belief was that offspring could inherit traits from their parents that their parents had actually acquired during their lifetime. And the classic example of that is the case of the giraffe's neck. So if a giraffe had spent all of its time working hard and reaching higher and higher to reach leaves, during the lifetime of that giraffe, the neck might actually grow a little bit longer because of the constant use, and that longer neck would then be passed on as a trait to the next generation. Another example might be that if I worked out really hard throughout my adult life and built up a really good set of muscles, that I could pass those great muscles on to my child. So Mendel set out to study inheritance in order to determine whether or not either of these assumptions were true, or whether there might be other rules and other principles governing inheritance. One of the experiments he conducted was to cross the tall plant variety with the short plant or dwarf plant variety. Now, if the first hypothesis we just discussed were true, meaning that offspring would exhibit traits that were a blend of the parental traits, what would you expect the offspring of these two plants to look like? You'd probably expect that the offspring would be about medium height, right? Somewhere in between tall and short. But that's not what Mendel got. And many of you probably remember this from grade 12. When he crossed the tall with the dwarf, all of the offspring were actually tall. So he concluded from this experiment that when you cross plants with two different traits for the same character, one of those traits will dominate over the other trait. Because these offspring are the result of a cross between two pure breeding varieties, it's considered to be a hybrid. And because the hybrid nature of it is with respect to only a single character, which is the height, it's known as a monohybrid. Now, given that all the offspring from this cross were tall, that sort of begged the question of what happened to the information in the short plants that made them short. What happened to that trait? Was it gone? And this was a question that Mendel asked as well. And so to try and help answer that question, he decided to cross these tall offspring with each other and see what happened. So he crossed the tall offspring with other tall offspring in order to see what the next generation would look like. This cross, because it's crossing a monohybrid plant with another monohybrid plant, is known as a monohybrid cross. And when he made this cross, he found that some of the offspring were tall, but some of them were short, and the ratio of tall to short was roughly 3:1. So the short trait came back again in the next generation, and that told him that although the short trait in these monohybrids was being masked or was not visible, it was still there and it was still intact, and it was still passed on to the next generation. And when he did similar experiments looking at other characters for which there were two different traits, for example, these shown here, he obtained the same results. In each case, one of these two traits would be dominant over the other, but the other hidden trait would return or be visible again in the offspring that resulted from the monohybrid crosses. The ratios were always about 3 of the dominant trait to 1 of the non-dominant trait or the recessive trait. When he thought about how he might be able to explain these results, he decided that the simplest explanation was the following: that each individual has 2 hereditary determinants for each character. In the cases where 2 determinants for the character in a plant would be different, one of those determinants would be dominant over the other, and it was the dominant determinant that was going to be visible. And a third conclusion is what is known as the principle of segregation, which states that 2 determinants of a character will always be segregated during gamete formation, with each gamete receiving 1 of the 2. Now, Mendel knew nothing about DNA and genes and alleles, but we now know that what he referred to as a character, such as plant height, is due to a single gene, and the different forms of those characters that he called traits are due to the different alleles for that gene. So let's explore another of Mendel's characters now and try to explain his observations in terms of modern molecular genetics and cell biology. Mendel also looked at plants that had seeds with different shapes. Some varieties produced seeds that were round, and other pure breeding varieties produced seeds that were wrinkled. We now know that this character, meaning the shape of the seeds, or the shape of the peas themselves, since the seeds are the peas, is determined by a single gene, and that gene has 2 alleles, one that codes for round seeds and one that codes for wrinkled seeds. When Mendel crossed a pure breeding variety that always produced round seeds with a pure breeding variety that always produced wrinkled seeds, and this generation would be known as the parental generation, they always produced offspring that were all round. These round offspring here are known as the F1 generation, or the first filial generation. So the round allele is dominant over the wrinkled allele. Looking at this cross using genetic notation then, we have a character or a gene that is responsible for determining the seed shape. There are 2 alleles. One allele codes for round seed shape and one allele codes for a wrinkled seed shape. Now Mendel noted that each individual would have 2 determinants or 2 alleles coding for seed shape. So the pure breeding round variety plants would have 2 round alleles, and the pure breeding wrinkled variety plants would have 2 wrinkled alleles. Mendel also noted that these 2 determinants or alleles would be separated or segregated during gamete formation. So that means that when this round plant generated its gametes, some of them would get this round allele to the left-hand side, and some of the gametes would get the round allele on the right-hand side. That means, though, that all of the gametes produced by this organism would all have the round allele. Similarly, in the plant that had the wrinkled alleles, half of the gametes formed would receive the wrinkled allele on the left, and half of the gametes formed would receive the wrinkled allele on the right. All of the gametes, however, would have the wrinkled allele. When you cross these two plants, or the parental generation, to produce the F1 generation, they are all round, but each one of these plants would receive one round allele from one parent and one wrinkled allele from the other parent, and so all of them would have both a round and a wrinkled allele. Now, you may remember that when you have individuals that have two alleles of the same type, as in the case of the parental rounds and the parental wrinkles, these are known as homozygotes. And the round, because it has two of the dominant alleles, are known as homozygous dominant plants. The plants that have two wrinkled alleles are known as homozygous recessive. Individuals that have one of each type of allele, as is the case in the F1 generation, are known as heterozygotes because they have two different kinds of alleles. Now, what if you cross these F1 hybrids with each other? Again, this is known as the monohybrid cross. Well, during gamete formation, these two alleles, the round and the wrinkled allele in each one of these hybrids, will be segregated so that each gamete receives only one of the two alleles. But what is the mechanism for separating these two alleles during gamete formation? Well, think about that for a minute. We've got an individual here. That individual has two alleles that code for seed shape. If there's two alleles inside that individual, then it's pretty safe to say they're probably diploids, right? So what process are we talking about if we're talking about separating alleles for the same gene in a diploid organism in order to make sex cells? I hope you're thinking about meiosis. So when we talk about the segregation of alleles in order to make these gametes, we're really talking about the process of meiosis. Now, the gene for seed shape is found on chromosome 5 in the pea plant genome. So this is what chromosome 5 would look like in one of those monohybrids that we were just talking about. If this was a germline cell, then it would be getting ready to go into meiosis. So at some point during interphase, prior to the beginning of meiosis, these chromosomes would replicate and produce sister chromatids, as shown here. One of the chromosomes has the round allele, and one of the chromosomes has the wrinkled allele. This cell would now go through meiosis. In the first division, the homologous chromosomes, and here we've got both of the chromosome 5s, would pair up in prophase 1 and would separate in anaphase 1 to produce two daughter cells, one of which received the chromosome with the round alleles and one of them that received the chromosome with the wrinkled allele. In the second meiotic division, the sister chromatids would be separated and we'd end up with four daughter cells, half of which contained the round allele and half of which contained the wrinkled allele. And what I've just described is essentially Mendel's principle of segregation, where the two alleles of the gene are going to segregate with each entering a different gamete, and it happens during meiosis. Now, if you're clever, you might be wondering, what if there's crossing over? How does that affect the outcome in the daughter cells? Well, let's have some crossing over. We'll start again at the top here with the germline cell. It's a heterozygous germline cell. It has both a round allele on one of its chromosome 5s and a wrinkled allele on the other. These chromosomes are duplicated during S phase, and then following the G2 after this S phase, they go into meiosis. And let's assume that during prophase, after these two chromosomes pair up, there is an exchange between sister chromatids that results in one of the round alleles being passed on to one of the wrinkled allele sister chromatids. So now we've got a mix. Rather than round, round, wrinkled, wrinkled, we now have a round and a wrinkled on one chromosome and a round and a wrinkled on the other chromosome. At the end of meiosis 1, each of the daughter cells has still 2 sister chromatids per chromosome, but in fact has 2 different alleles because each sister chromatid actually has a different allele for the seed shape gene. These undergo meiosis 2, and you end up with 2 gametes or daughter cells that have the round allele and 2 gametes or daughter cells that have the wrinkled allele. So the outcome, 50-50, is still the same. Now let's get back to our question of what happens when you do a monohybrid cross. Can we actually predict what Mendel would have seen based on our knowledge of meiosis and fertilization? And the answer is sure we can. Let's remember that the outcome of meiosis in both of these monohybrid organisms is going to be 50% of their gametes will have the round allele and 50% of their gametes will have the wrinkled allele. And we're going to cross them with each other. In order to predict the possible outcomes, we're going to use what is known as a Punnett square, and I'm sure most of you are familiar with Punnett squares. You simply create a grid, and you should have a row for each of the possible male gametes or sperm, and you should have a column for each of the possible female gametes or eggs. So in our case, we're crossing 2 heterozygotes. So in both cases, they have 50% of the dominant allele and 50% of the recessive allele. So now we place the possible alleles for the sperm beside each of these rows. So one of the rows will have the round allele associated with it, and the bottom row will have the wrinkled allele. These are the 2 possibilities for sperm. And now we, across the top here, we put the possible alleles that can be found in the female gametes or the eggs. Half of them will have the round allele and half of them will have the wrinkled allele. Inside the boxes now, you bring these alleles together. So we take the round allele from the father and the round allele from the mother, and we put them together here. We take the round allele from the father here, and then the wrinkled allele from the mother, and we put them together here, and likewise in the other boxes. Each one of these boxes shows the possible genotypes for the offspring, where the genotype refers to the alleles that the offspring will have for this particular characteristic. So these offspring will have 2 round alleles. They'll be homozygous dominant. The genotype for these individuals will be heterozygous. These individuals in the third box at the bottom left will also be heterozygous, and the individuals in the bottom right box will have a genotype which is homozygous recessive. Each one of these boxes, because there are 4, represents 1/4 of the offspring. So this tells us that 1/4 of the offspring will be homozygous dominant, 2/4 or 1/2 of the offspring will be heterozygotes, and 1/4 of the offspring will have a genotype which is homozygous recessive. We can also translate these genotypes into phenotypes, meaning what is actually visible. We know that both the heterozygotes and the homozygous dominant will have the dominant phenotype, and they will have round seeds. And the homozygous recessive individuals will have wrinkled seeds. This gives us a ratio for round to wrinkled seeds of 3:1. This is exactly the ratio that Mendel saw when he did these crosses. So just to review for a minute all of the terms we have used in this presentation. Now, we've been talking about genes and the behavior of genes, and we've been relating them to the phenotype, which is the visible trait, the trait that we can see. So what then is giving rise to these different phenotypes? In other words, can we explain the different phenotypes we see in round and wrinkled in terms of what we know about what genes do and what gene products are? So pause this video, read this question, and see if you can answer it. Let's start thinking now about what alleles are, what genes are, and what genes produce. What exactly is the gene for seed shape coding for? If you think about a pea, which is a seed, it contains a lot of starch because starch provides energy to the growing embryo. In pea seeds, starch consists of amylose and amylopectin, shown here. And the only difference between the two is that amylopectin has branched chains of sugars, not just linear chains. Amylose consists only of linear chains of glucose. It turns out that the R gene codes for an enzyme that makes amylopectin. In other words, it can give rise to these branched chains off of a linear chain. So what's the difference between these two alleles? The allele that codes for round and the allele that codes for wrinkled. If you remember what alleles are, they're different forms of a gene. And if they're different forms of the same gene, then both alleles must be coding for that same enzyme. So why the difference in phenotype? Well, let's take a look at what the difference is between the round allele and the wrinkled allele. The two alleles are coding for different versions of this enzyme that makes amylopectin. The round allele codes for an enzyme which is fully functional, and that means it's able to synthesize amylopectin. The wrinkled allele codes for a different version of that gene, and in this case it's a different version because somewhere in the coding sequence there is a difference in gene sequence, which results in the creation of a stop codon much earlier in the protein. This results in a truncated protein being made from this allele, and that truncated protein is a non-functional enzyme. So the round allele codes for the functioning enzyme, and the wrinkled allele codes for a non-functioning enzyme. So we have our two alleles now, and we now understand what the gene codes for, and we understand what the difference is between the two alleles. How does this give rise to the two phenotypes of round seeds and wrinkled seeds? Well, in both of the pea seeds, whether it's a homozygous wrinkled or a homozygous round, they both contain amylose. However, they don't both contain amylopectin. The homozygous round seeds contain normal amounts of amylopectin. The homozygous recessive seeds contain no amylopectin. And it turns out that in these seeds, not only is there no amylopectin, but there are large amounts of the disaccharide sucrose. And this large amount of sucrose means that there's more solute inside these seeds. So seeds that are homozygous recessive tend to have a larger amount of solute inside them, and they retain a lot more water initially. So they sort of swell up. When these peas mature, they lose the extra water and they dry out. And as the water leaves these peas, it causes them to wrinkle up. This is what gives rise to the wrinkled phenotype. So this explains the phenotype difference between homozygous round and homozygous recessive. But what about the heterozygote? What is happening there? Pause the video and answer this question. Well, to answer this question, you have to recall what you know about gene expression. So here we have two copies of the seed shape gene. One of them is the round allele, and here we have the wrinkled allele. These two alleles are identical. They are the same gene, except for a difference in sequence somewhere in the coding region that results for the wrinkled allele in an early stop codon being made in the messenger RNA. This means that the rest of the gene is the same, and that includes promoter regions, promoter proximal elements, enhancers. In other words, the regulatory parts of the gene are the same. So if the transcription factors for this gene are present in the peas in early development, they should be able to turn on both genes, right? Because the regulatory regions of both genes are the same. Does this make sense? Now try and answer this question. Again, pause the video. So think about this. You've got both alleles. They have the same regulatory region. They should both be turned on inside those cells. So what do you expect to see in terms of gene products? Well, if they're both being active, they're both producing messenger RNA, and if they're both producing messenger RNA, then presumably both types of messenger RNA are being translated, and that means you should have both types of enzymes. I would expect to see in those cells 50% normal enzyme and 50% abnormal enzyme. So we've now talked about Mendelian genetics, and we've connected it to what we know about modern molecular genetics, the nature of DNA, the nature of genes and their alleles, and how they give rise to proteins. And you'll notice that we're starting to put together information from across the course. Enzymes and enzyme activity, proteins and protein structure, genes, gene expression, meiosis, Mendelian genetics.